good afternoon conference. Thank you, Mr. Chairman.
Добрый день. Спасибо вам большое, господин председатель.
Do you have a pointer? Ah, super. There we go. I'm Anthony Mossop. Thank you for the introduction. It strikes me, having listened to the lectures here this morning and the ones that I can see that are coming, and with reference to last year, one of the things I love about this conference is that everybody here, both the people witnessing and all the people lecturing, are fundamentally interested in challenging scientific misconceptions, in investigating.
Добрый день. Еще раз, меня зовут Антони Моссов, и я очень рад присутствовать здесь. И сравнивая сегодняшнюю конференцию с тем, что было в прошлом году, я могу сказать, что замечательно, что все, кто выступают, и те, кто слушают, они стараются подорвать те научные недопонимания, которые существуют, которые мы все наблюдаем сейчас.
In investigating across multiple fields, we're seeking truth. With good science, thanks to GDV and BioWell.
Мы все ищем правду, и благодаря BioWell сегодня мы можем сделать ее более достоверной, действительно научной.
So I'm going to introduce our talk about Phytobiophysics with regard to its interrelationship with BioWell by looking at an investigation into the energetic properties of Phytobiophysics using the BioWell water sensor.
Я бы хотела поговорить о качестве фотобиофизики, фитобиофизических формул и совместно с BioWell и с использованием водного сенсора.
Now, in order to do that, just like Mr. Narayanan, I think it's expedient that I just introduce a little bit the concept of phytobiophysics before I start the actual investigation that we did.
И сначала я бы хотела представить понятие фитобиофизики, прежде чем мы двинемся дальше.
So, energy medicine is an evolving viewpoint of health and illness that takes into account all the many forms and frequencies of vibrating energy that contribute to the multidimensional human energy system.
Энергетическая медицина – это точка зрения, которая развивается сегодня о здоровье и заболеваниях, которые принимают в учет все формы и все частоты вибраций энергии человеческого тела.
It's based on scientific principles that all matter vibrates to a precise frequency and that by using resonant vibration, balance of matter can be restored.
И таким образом мы принимаем во внимание ту идею, что вся материя, она вибрирует с определенной частотой.
And in fact, these are basic principles that we're all using rather than just principles of phytobiophysics. It's also, do you want to translate that?
This one.
Don't worry. It's also an approach to the diagnosis and treatment of illness based upon the idea that we're all unique energy systems.
И в медицине в этой подход к диагностике и к лечению заболевания таков, что мы все являемся уникальной энергетической системой.
So some of the founding fathers of the science behind what we do and what we have theorized with regard to phytobiophysics, we go back to 1923, and Alexander Gurevich discovered the ultra-weak photon emission from living systems. I'm sure many of you are already familiar with some of his work and concepts.
Я думаю, что многие из вас знают понятия и разработки, которые были совершены Александром Гуревичем в 1923 году. Он был основателем фитобиофизики, и он открыл эмиссию фотонов.
So, in fact, we don't need to read out the whole lot here, but it was to do with his initial deductions on the radiation emitted by plant roots. Mitogenic radiation is what he termed it as, which we now know as. Where's the pointer on this? It's the red button. Ultra-weak photon emission or biophotons.
Не будем читать биографию целиком, но главное, что он занимался исследованием, извлечением света, то есть радиации растений.
One of his two remarkable properties that he discovered I find particularly interesting, and that is that although the light, the bio or the ultra-weak photon emission cannot reach inside all of the cells, you get this amazing secondary radiation from that cell. In other words, the information that's transmitted, even though it's light and it can't enter into the cell, can then be transmitted by a secondary means.
И интересно то, что он отметил, что несмотря на то, что свет может проникать через все клетки, информация может проникать благодаря вторичной радиации, вторичному излучению.
In certain, just go back one, in certain modern developments, scientists have begun taking interest in ultra-weak photon emission from biological tissue, and they've differentiated ultra-weak photon emission from light emitted, for example, by bioluminescence, and systems where light is initiated secondary emission photons.
И сегодняшние исследования, тоже исследования 60-х годов, исследования над тканью биологической, и также были выявлены вторичные излучения.
Again, here's someone that will be very familiar to many of you, Fritz Albert Popp. He found that all cells emit tiny packets of light, which he called biophotons. Now, the truth is that we know that all matter emits photonic energy, that all matter is energy, but I like the term biophoton because he's really talking about the light emission from living cells.
Многие из вас знают этого ученого, господин Поп, и все мы знаем, что излучение происходит не только от биологических веществ, но и от любой материи. Но, тем не менее, мне нравится название биофотон, эмиссия биофотона.
Light is constantly being emitted and transmitted by DNA molecules, and biophotons create a dynamic, coherent web of light.
Свет постоянно находится и передается с помощью ДНК и в ДНК.
Okay, it's also a system that could be responsible for chemical reactions within cells, cellular communication throughout organism, and overall regulation of the biological system, including embryonic development into a predetermined form.
И мы можем увидеть связь между всеми системами человеческого организма, химическими реакциями и коммуникацией внутри органов и организма, и регуляцией биологической системы в целом.
In fact, there's much alternative research into these areas, and some of the research, for example, is beginning to demonstrate that rather than being a chemical basis for smell, it is actually vibrational.
Существует множество исследований альтернативных сегодня. Одно из них, например, говорит о том, что запах — это не химическая реакция, а вибрационная реакция.
Very briefly, we're within Phytobiophysics particularly interested in the electromagnetic spectrum of the rainbow, i.e. that which our cells emit and that range of electromagnetic energy which make life possible on this planet.
В фитобиофизике мы заинтересованы в исследовании излучения клеток и магнитно-электрического поля.
So, Dr. Voll, again, he's been mentioned today. A lot of these people get mentioned again by all of us because they are the forerunners. But fundamentally, Dr. Voll's EAV acupuncture resistance testing the acupuncture points vibrate on specific frequencies and emit energy on a constant level when healthy.
Доктор Волл также известный ученый, который исследует электроакупунктуру и сопротивление этим исследованиям.
This is a galvanometer, which is the testing method. This device was originally designed to measure electrical frequency through telephone wires and telegram cables, I believe. So it's interesting how technology can cross over. But of course it has one major drawback, and that is it is not an objective piece of scientific equipment, rather it is considered subjective.
Интересно, что вот этот вот прибор использовался в телекоммуникациях при исследовании телеграфа и телефона, но теперь он переместился в нашу науку.
I don't have very much written here because I think, again, all of you are going to be aware of Semyon Kurlian's work. I understand from Professor Korotkov that there was much research behind gas discharge visualization or the bioluminescence effect even before Semyon Kurlian, but of course this made it famous worldwide.
Ну и все вы знаете исследования Кирлиана и работа господина Короткова основана на методе Кирлиана и ГРВ также на нем основывается.
This was a particular picture taken with a Phytobiophysics client. That's the before picture. It's a healthy child, but they were suffering with immediate emotional stress at that point. And that photograph was taken 5 minutes before administration of one of our formulas. And you can see that there's been quite a transformation. That's only 5 minutes difference between the two samples.
Вы видите здесь две фотографии, разница во времени между ними всего 5 минут. Первая была сделана перед тем, как с ребенком была проведена фитобиотерапия, и сразу же после нее.
What discussion about, I'm doing water analysis, so of course I kind of have to mention Matsuro Emoto's work, and of course phytobiophysics is a healing philosophy fundamentally connected with flowers, so I just think it's nice to show these crystals and their perfect formation, which even closely resembles the flower essence from which they came from.
Я бы хотела поговорить также об исследованиях воды, которым, конечно, невозможно избежать. И фитобиотерапия связана с цветами, и вы видите, что здесь кристаллы воды очень сильно напоминают цветы.
However, again, although this is wonderfully demonstrative, I understand that in order to get some of these perfect crystals, he has to repeat it many, many times and doesn't always get perfect crystals. In other words, it's very demonstrative, it's wonderful, it gives us an indication that something's going on, but it is not definitive proof.
Для того, чтобы получить такие идеальные кристаллы, требуется большое количество работы, но это возможно.
Konstantin. Here he is, and there he is there. Now this photo we like because it demonstrates that he's not just agile in the mind, but that he's agile in the body as well.
Здесь мы видим фотографию Константина Короткова, и видно, что он светил не только душой, но и телом.
It's the perfect opportunity just to mention, of course, because what we're using is the gas discharge visualization technique via the biowell, and of course without Professor Korotkov, none of this would be possible.
Мы используем систему ГРВ в наших исследованиях, в нашей лаборатории, и без Константина Короткова это бы не было возможно.
So there is everything. Everyone recognizes this. Not everyone will have seen the bio-water sensor, and I'm delighted actually that you mentioned it earlier on this morning in terms of its possible uses with a thought experiment. I think that's quite handy for me because it sort of already introduced it to some of you.
Ну и вы узнаете аппарат BioWell в данном случае с водным сенсором.
So, go on to the methodology of this investigation. And what we're looking at is in order to investigate the properties of phytobiophysics formulas with the BioWell water sensor, we determined we'd need to try to control the experiment in the following ways.
Здесь я бы хотела рассказать о методологии. Мы рассматриваем фитобиофизические формулы и их влияние вместе с BioWell и водным сенсором.
Number one, we needed to select and use a stable water as a base for the tests.
В первую очередь мы отбираем воду для исследования.
I'm not going to discuss water in depth today because, of course, there are many people who are going to do that or have done that before. A lot of information, which is very exciting there.
Я не буду много говорить о воде, поскольку на нашем конгрессе и так много ученых, которые рассказывают про воду.
Very simply, to use the same shape of receptacle for the test in a temperature-controlled environment.
Мы проводим исследования в условиях контролирования температуры.
And to remove as many other influences from the testing area while the tests were being conducted, i.e. turning off other equipment, running the test in offline mode, and removing the tester from the environment as rapidly as possible after initiating the test. All the same parameters, in fact, that Professor Korotkov mentioned earlier on this morning.
И мы используем все те параметры, которые профессор Коротков упоминал сегодня ранее, то есть мы исключаем все наличие любых приборов, которые могли бы влиять на прохождение исследования.
Now we all know that water can receive information. We all know that it can hold it, that it has a memory, and that implies, of course, that it can also transmit it. So it's actually quite important to remain mentally mentally objective, and try not to hold preconceptions about what results might be produced while the test was in progress.
Мы все знаем, что вода не только сохраняет информацию, она ее и передает. У воды есть память, поэтому очень важно, проводя исследования, не быть предубежденным с самого начала.
Now, we conducted this test on 20 separate tests on each of the cases that we investigated for at least 20 minutes. What we actually investigated was 5 of our formulas and a placebo.
Мы проводили исследования с 20 препаратами, и в течение 20 минут у нас было 20 исследований с различными, с 5 препаратами, с фитопрепаратами.
We chose to initially analyze only the energy data provider, which is a combination of area intensity. And we chose to select this after 10 minutes of testing time from the starting point at which we started analyzing the data. That was really to allow the pilules, which is the form we transmit the formulas in, to be fully dissolved and to allow the settling of the testing data.
Мы использовали препараты, которые полностью растворялись в воде за 10 минут.
So this is just an example of the data generated from the BioWell water analysis. I don't know if any of you have looked at this data in the CSV capacity, but just to make it clear to you that this is every 5 seconds you get a readout for area, intensity, energy, and standard deviation. And in the 20-minute tests we get 250 of these columns of data for each test.
И здесь вы видите результаты, те данные, которые мы получали во время исследования. Каждые 5 секунд новые данные, то есть за 20 минут мы получили более 210 такого рода колонок.
So there's a huge quantity of data to analyze, and like many people who enjoy researching but also have other work obligations, I found time was my main limiting factor.
Это огромное количество данных, как вы понимаете, и для меня основной проблемой было найти время, чтобы все ее проанализировать.
Just to give you an explanation of those parameters, again, just to highlight them. Area is the number of pixels of the glow. These are rough parameters. Intensity is the average intensity of the glow. And energy in joules, the factor that we're using in our studies. The area multiplied by the intensity multiplied by a correction coefficient.
Хотел бы прояснить несколько терминов, которые мы используем. Площадь — это количество пикселей свечения, интенсивность, то, что вы знаете, и энергия. Энергия — это умножение площади на интенсивность на коэффициент коррекции.
So our initial test, this is actually just an excerpted photograph of the lines. This actually, sorry, it seems to have cut off the time here, but this was right at the beginning. We didn't start analyzing data over until this point, but you're looking fundamentally here at 3 groups of water. We only conducted for the water about 5 or 6 tests on each sample of water, but you can very clearly see how they are banded together in groups. 1 group, 2 groups, 3 groups.
Здесь вы видите исследования над 3 типами воды, и мы проводили всего 5-6 тестов для каждого вида воды, но вы видите, как четко они сгруппированы на графике.
What we did with that data after 10 minutes, again, because we wanted to be consistent, is that you can see very clearly that what we did is we took a mean average of the data collated from the multiple tests in order to give these individual plot lines at these certain time intervals.
Мы решили сделать медиану. В каждом исследовании на графике вы видите разные типы воды, и разница во времени была 10 минут.
Now that is a very simple statistical technique, and everyone will be aware that when you have very disparate averaging, very disparate data that's very high or very low, it can skew those results. But that's why I think it's important just to point out there that those data groups were very much in individual bands anyway.
Мы предоставляем медиану, но вы видите, что в любом случае данные были сгруппированы по каждому виду воды очень четко.
So the first group that we tested with was tap water. Now you will have seen the disclaimer with regard to the water analyzer. It doesn't test biological purity. We're looking at different information.
Следует понимать, что мы смотрим не биологическую чистоту воды, но разного рода информацию. И в первом случае оранжевым цветом отмечена вода из-под крана.
What I can tell you is that the tap water from where we live, although biologically pure, is very toxic. It has the highest nitrate levels in Europe, which of course has been linked to cancers.
Несмотря на то, что вода из-под кранов, из того места, где я живу, считается очень биологически чистым, на самом деле она токсична, и некоторые исследователи связывают характеристики воды с раком.
Now, we can see that the tap water line is quite definitely down here. The next group of water that we actually tested was this, which is reverse osmosis filtrated water.
Второй тип воды – это фильтрованная вода.
And what we did then is we actually added to this water, because obviously it takes out all of the mineralization, we added to it Adia Clarity, which is an ionic sulfate mineralization.
Мы добавили Adia Clarity во второй тип воды, которая насыщает воду минералами. Прежде вода была полностью отфильтрована от каких-либо минералов.
Concept being that we are adding back to the water what we happen to have stolen from the tap water by reverse osmosis. The trouble with tap water is it contains all the chemicals. It also contains all the micropharmaceuticals as well.
В воде из-под крана огромное количество химических элементов и минералов.
Just as a quick point of interest, Adia Clarity website, which sells their particular ionic sulfate minerals, which comes from black mica, they have direct links on their website to presentations given by Professor Voykov and indeed Professor Konstantin Korotkov as well.
И вся информация по составу Adia Clarity находится на веб-сайте, вы можете посмотреть.
So it shows how good information travels.
И это является доказательством того, как информация распространяется.
We selected the Adia Clarity water as the water to do our tests because it had very stable data. We knew it had been remineralized and we knew it didn't have any chemicals in it.
И у нас есть достоверные данные по тому, какие элементы содержатся в этой воде, именно поэтому мы выбрали ее для исследования.
So the Phytobiophysics heart power formulas, I'm not going to go into them in depth. We've got brochures out there for you, but fundamentally they are 5 formulas linked to the 4 quadrants of the heart or the 4 heart valves and one that we call heart center point there in the middle.
Вы видите, это был наш логотип фитобиофизики.
Yeah. Very quickly, what is phytobiophysics? Phytobiophysics is a philosophy of understanding and treating energy imbalances in the human organism.
Что же такое фитобиофизика? Очень коротко, это то, каким образом мы понимаем и лечим дисбаланс энергии в человеческом организме.
And its primary method of treatment is via flower essence combinations that resonate on specific frequencies.
И основной метод – это цветочная эссенция, которая резонирует на определенной частоте.
Now we have over 1,000 different formulas currently made up of countless individual combinations of flower essences. We're only looking at 5 here, and that took over 40 hours of testing alone.
Мы создали более 1000 формул на данный момент, созданных из цветочных эссенций.
So the combinations have been painstakingly put together over many years of in vivo clinical research and treatment according to the principles of Vol. And of course you know that those same principles have been used to combine with GDV technology to create the BioWell program.
И мы использовали принципы, которые in-vivo клинических исследований, которые были созданы господином Волом, а также использовались при создании ГРВ.
Now, one of the most common terms used by current mainstream medical thought when referring to all types of vibrational medicine, and indeed most holistic techniques, is that it's all placebo. Let's put to one side for the moment the fact that they can demonstrate that placebo pills are better for treating certain medical conditions such as asthma and depression anyway.
Что мы слышим чаще всего, когда мы говорим о такой области, как фитобиофизика, то, что это плацебо.
Thus, it is incumbent upon us to commence the process of investigation using objective scientific equipment, the GDV.
И таким образом мы решили начать исследование с помощью ГРВ для того, чтобы доказать обратное.
Now these are actually, there's the water sensor, it's just to show you, these are actually the placebo tablets that we use in Phytobiophysics. They are actually, well, I'll sip on because it says here. The pills used are indeed a pharmaceutical grade sucrose placebo pill. However, they've been coated with the relevant essence combinations in our GMP registered laboratories, using a method similar but not identical to classical homeopathic techniques.
Вы видите здесь таблетки, которые являются плацебо, но с помощью специальных технологий они были покрыты эссенциями и фитобиофизическими формулами.
So this is not simple flower essence therapy like Dr. Bach. And the main reason they are in a pilule form is so we can avoid the need to hold the tablets in alcohol, and so that they can, according to our understanding, hold the frequency accurately without undue external influence altering the complex interactions, and also that the amplitude can be maintained by the crystalline structure of the sucrose.
Это отличается от предыдущих технологий, поскольку мы можем следить за частотой использования этих препаратов и за частотностью внутри этих препаратов и их влияния.
Now, in truth, it might be better to put them into salts, but people don't like taking salt tablets repeatedly throughout the day.
Можно поместить их в соль, но людям не нравится на самом деле есть много соли.
So that's just a picture at this point of our 5 heart power and the placebo. So that's 120 tests. It reminds me of making Sunday lunch for my family. It takes 5 hours and it's all over in 5 minutes.
Напоминает немножко это исследование на обед с семьей по воскресеньям, когда занимает 5 часов подготовки и 5 минут и все закончилось.
I just want you to see generally the range from down here from about 1 to about 5, but obviously this one is particularly aberrant, and I want you to just look at that pattern because we're going to come back to that in a minute.
Я бы хотела, чтобы вы обратили внимание на этот график, посмотрели внимательно. Здесь вы видите HP 1,5 и в сравнении с плацебо.
So obviously you can't make out anything very much there at all apart from the top and the bottom. I'm the same, so let's start trying to tease that data out.
Давайте попробуем рассмотреть данные подробнее.
So this is just an excerpt of the example of the collated data taken from the 20 tests on the same formula. That gives us the relative plot points taken after a point starting at 10 minutes. It's just an excerpt of that data because it continues and continues for hundreds of levels.
Пример теста, проведенного 20 тестов, проведенного с одной и той же формулой в течение 10 минут.
So the first formula that we looked at, Heart Power 1, there, again, it's in the brochure. It's the heart center point. And I just wanted to show you here, Heart Power 1 above placebo. Now, I know very clearly that this orange line here, and one of the reasons we started after 10 minutes, this orange line here is the superior range of the placebo test. But look, we didn't start assessing the data until this point when all the data had started streamlining down into relative bandwidths.
И на этом графике вы видите, что HP1 находится над плацебо. Вот эта оранжевая линия – это пограничная линия плацебо. Все, что ниже – это плацебо.
So just visually, we can see that there's actually quite a lot of data above the most superior, which was almost aberrant, of this test of the placebo data down here.
Вы видите данные исследований плацебо, вот эти оранжевые, но над ней большое количество информации.
At this stage, it's just demonstrative, right? So Heart Power 2 was the next one that we did. And again, you can see that same orange line, but here again, you can see that the data line has generally moved up, so that this is all Heart Power 2 versus placebo up here.
HP2 и плацебо в данном случае вы видите, что вся информация двинулась выше. Оранжевая линия все так же плацебо.
I might just point out again that the disparity in the information provided on the energy readout is much greater at the beginning of the test and much more combined after 10 minutes of stabilization. That's the point at which these sucrose pools coated with the essence have dissolved.
И разница в информации в начале теста, вы видите, намного шире, позже она объединяется через 10 минут.
Right, you might think that this is a funny picture to have in my presentation because suddenly we've got 2 dogs. This is actually my dog Flora and this is my puppy Poppy.
Это моя собака Флора и щенок Poppy.
Poppy's only a baby, she's only 3 months old, so I excuse her behavior, but you'll see why.
Ему только 3 месяца.
So with Heart Power 3, we had a problem. Here it was. It's that data line which I showed you. It's the most superior and the most aberrant data. Unbeknownst to me, Poppy had got into my lab. when I was doing this data test.
Зачем же была предыдущая фотография? На следующем исследовании HP3 вы видите, что данные меняются сильно. Что же случилось? Щенок бежал в комнату.
Again, we can see down here the placebo, the most superior line of the placebo test, the placebo grouping. And we got heart power 3, but actually what happened is Poppy got into my lab, Poppy made a mess in my lab, I discovered Poppy, kicked her out, and then I had to tidy the lab. And it took a long time for this data line, but it did eventually come back down into stabilization with the other HP3 data.
И снова оранжевая линия — это плацебо. Вы видите все, что выше в потоке информации. В начале эксперимента забежал щенок в комнату, затем он беспорядничал там, потом я заметил это, и дальше вы видите, что происходит стабилизация.
Apart from proving that puppies can be a nuisance, it does actually also demonstrate the sensitivity of water as a medium of receiving information and the ultimate care that we have to take in doing the tests accurately as possible without these influences.
И в данном случае это не просто помеха и шум в данном исследовании, а это показатель того, насколько вода является чувствительной, насколько осторожными мы должны быть при проведении тестов.
So moving on quite quick, heart power 4 is another one of the formulas, and when we look at that data, then heart power 4 over placebo, I believe that heart power 4 was very closely linked to this secondary part. That's actually the top line of the placebo data, but heart power 4 seemed to exhibit the properties of this bandwidth immediately above that one. So it's very close together, but of course our formulas are meant to be vibrating on different frequencies.
Вы видите, в данном случае HP4 плацебо оранжевой линии находится довольно высоко, но несмотря на то, что линии и частота кажутся схожими, на самом деле наш препарат и плацебо действуют просто на разных частотах.
So, and heart power 5, again, then we've got here the superior line of the placebo test, and we've got the data very nicely bunched together for heart power 5. You can see it's very concentrated in this central area here.
И HP5, результат оранжевой линии — это плацебо, и наверху вы видите конденсированную линию нашего препарата.
And this is just really to demonstrate that what I actually did here is I took the two heart power formulas which were closest together in their ranges of data, and actually fundamentally what we found is that the beginning point of one set of data overlapped with the end point of the other set of data. So from about here down and from about here up, we're looking at the two different sets of data, and there is overlap.
Мы решили сравнить HP3 и HP2, и мы видим, что на графике, там, где заканчиваются данные HP3, начинаются данные HP2. Это интересно, что получается, что они пересекаются.
And indeed, we could see that in this data set as well. But what we did ultimately is we had to collate all those multiple reams of data. Remember, we're just looking at one. This is just an example of the excerpted data, but this down here is our placebo. The average readings of the placebo test taken over 20 samples. This is the time it starts after 10 minutes.
Здесь проводится комбинированный анализ результата нашего исследования. Вы видите, плацебо находится вот между 1,2 и 1,4 синяя линия, и все, что выше, результаты наших препаратов.
And then obviously we can see that the other heart power formulas are exhibiting specific lines which are all different to each other and all different to the placebo.
И вы видите, что линии все разные, все отличаются, и они отличаются от плацебо тоже.
It's interesting to note that the placebo actually, because what happened is that the water that we had structured with Agia Clarity after reverse osmosis was having a readout of about here, about 2.2. And that dropped right down with the sucrose simple solution to 1.256.
Интересно, что исследование с той отфильтрованной водой, которую мы использовали, было на уровне 2,2, но потом раствор сахара снизил ее практически до нуля.
So these lines are also exhibiting considerably different information to the water as well.
Таким образом, мы видим разную информацию, хранящуюся в воде.
Very briefly, in conclusion, the data provided shows that the energy readings of the heart power formulas differ substantially from those readings provided by solely the placebo pills in the chosen water.
В заключение хочется отметить, что данные, которые выдают hard power формулы, сильно отличаются от данных, которые дают плацебо.
The heart power formulas register specific bandwidths of data that often originate and terminate within the bandwidths of other data immediately below and above that bandwidth, but they rarely extend into the bandwidths of other data groupings.
Те данные, которые мы получаем от формул hard power, часто перекликаются, но никогда не накладываются одна на другую, одна начинается после другой.
And that the highest average energy readings provided are at least double those given by the placebo, which I think is quite definitive.
И разница, которую мы видим, отличается с плацебо в 2 раза.
Points of interest for consideration and further work. Well, of course, as we already discussed, that was a very time-expedient method of simple statistical observation, but more in-depth statistical analysis does need to be conducted on the mass of data gathered.
Мы предоставили очень простой статистический анализ, и стоит отметить, что необходимы дальнейшие исследования в этой области.
And there is a need to cross-correlate the findings with the other, within the energy parameters with the data analysis of the other parameters as well.
Кроме того, необходимо провести корреляцию с нашим исследованием и другими исследованиями в этой же области.
Finally, from my part, the other 1,000 or so formulas from Phytobiophysics will need investigation, i.e. it's a lot of work to do.
Кроме того, более 1000 формул, имеющихся, также необходимо исследовать, что большая работа.
I think I calculated that just collecting the data is going to take me 4 months of solid work from 9 till 5 without a break every 15 minutes.
Я думаю, только собрать одни данные мне потребуется несколько месяцев работы с 9 до ночи.
I'm hoping with the new equipment I can cut some corners. Thank you very
Возможно,
much.
с новым оборудованием я смогу сократить время.